not always torsion. Another important thing that you should know about torsion. Torsion is different than coiling. When these animals, the gastropods, coil, is that right? That's called coiling. When the shell goes around, it's called coiling. But torsion happens only in gastropods. And what happens moving of mantle, twisting the visceral organ 90 to 100 degrees. Maybe I should draw this. The ancestors, maybe I do have it. I have a PowerPoint, I think I'll put it later on. Twisting the visceral organs 90 to 100 happens in villager stages. Anus, villager, again, I'm sorry, villager stages are for both of these guys. Both of these guys. And then anus opens to the mouth, and that's called fouling. We do not understand. Okay, here is the mouth of the animal is here, right? And of course, guess where is the anus? Right here too. And that is called fouling, where the mouth and anus are in a close proximity in any animal. Usually only happens in these animals. When the anus and mouth, again, complete digestive system. This is not like Hydra you study. They have complete digestive system. The mouth and anus, where the waste material comes, usually on close proximity. So it's called fouling. Again, evolutionary, your textbook might get into it. Evolutionary, what is the, why these animals did this? Why these animals wanted that the anus end up on top of their mouth? We really, really don't know. We do not have an explanation for it. That's one of the things, again, a lot of things in biology, we do not have. Your textbook, I don't know, tries to get into it. I didn't understand it. Maybe you, I don't think it makes sense what they explain. Coiling, again, is the coiling of the shell, and spiral winding may occur during the larval stages. This is a name of a subclass in early gastropod. Again, if I gave you, here we go. So, here is the mouth and anus, but eventually when the animals coils, the mouth and anus end up on the same area. Okay, that's where the early stages. The evolutionary wise, the bottom one is the evolution of these animals. They had a mouth and they had the anus. Most animals are like that. Most all animals are like that except these guys. But over a period of time, they evolved and they vented their digestive tract and eventually ended up like the modern gastropods we have, the mouth and anus in the close proximity. Again, we do not have an explanation for that. These were the early ancestors of the gastropods. Of course, everybody's like that, but the early ancestors had a 90 degree, that's what I said, 90 to 180 degree. That was a 90 degree digestive tract, and then eventually ended up with 180 degree, all the way back where the mouth is. Here's an internal anatomy of it. Don't expect you to know. We do have a model of it in the lab, but again, I think you have enough material to worry about. The three subclasses, I've just put them in there again, not to worry about them for lab practical, but periwinkle, I don't think we have it. Horse conch, we don't have them in the lab. Sea slugs, I don't think we have them, but helix, we have them. Limex, I don't think we have them. Physa, we do not have them. Let's go to the next class. Class by value. Pelicipoda. Another name for it is Pelicipoda. Freshwater mussels. Hatchet foot. That's what I mean by hatchet foot. Right here. It's like a hatchet. And then clams, scallops, oysters, shipworm. All of them we have in the lab. All of those specimens, we have them in the lab. Most are sedentary filter feeders. They move, but they move very slowly. And no head, no radula, very little cephalization. Open circulatory system, of course, just like this one. Hemocyanin is a pigment of colorless blood that these animals have. And nephridia near the heart, on the bottom of the heart. Heart is on top when you dissect. The heart is on top, and nephridia is right on the bottom of it. If you dissect carefully, you will see them. They both are brownish color when you dissect them. But if you are not like me, you're very patient, you're very careful, and you're good with your hand, you can separate them apart. Okay, they're covered by a connective tissue. But this is what I got. A lot of people ask me in the past, students, Amir, what's the difference between mussel and clam? If you read that statement, I got it from online, really, there is not much difference. The term mussel is used for several families of bivalve mollusks inhabiting lakes, rivers, and creeks, as well as internal area, intertidal areas along coastlines worldwide. So mollusks, mussels and clams are sometimes it's several families, you know, phylum, class, order, genus, family. You all know what I'm talking about. The taxonomic group, including most bivalves, referred to as clams. Same here. Shell, again, two shells. They are connected together by a hinge ligament. There is a ligament that connects it to them. Anterior and posterior adductor muscles. These are the muscles. That's why when you have a tough time right here, these are the anterior and posterior. This is the anterior, sorry, and this is the posterior. Where do you know the anterior? The way I say, by umbo. Umbo is the oldest portion of shell. So that's the anterior portion of the animal. The posterior portion of the animal is this one. So these are adductor muscles. So these two muscles are really, really they're tough. You cannot open the shell. So you have to, if you're going to dissect with your scalp, you have to cut these two in order to be able to open up the shell. And they work antagonistically. One contracts, the other relaxes, the other contracts, the other one contracts, so on. Umbo is the oldest portion of the shell, the 3 layers of shell. I think this is the second time. Pearl production, I will talk about it. This is Japanese oyster that makes pearl. I gave you the name of the genus. Melagarinia is the name of the organism. This is the only. Here we go. Foot is attached to visceral mass. We already talked about that. The edge of mantle folds are modified to form excurrent and incurrent siphon. Locomotion, blood is pumped. Blood is pumped right here. Blood is pumped to the foot. So foot goes to the sand or rock or what have you and push the animal, of course very slowly, push the animals, and then they do it again and again. That's one type of movement. The other type of movement, they open up, they have the ability to open up the shell like this and squirt water. A lot of water goes in there. Squirt water out so they can move away from the predator. If a starfish is attacking, starfish love to eat these guys. So they have the ability to do that, but how much they can do that, not often. The way they do move typically is this way, by their foot. But how? Not much. They cannot do this all day. Okay. Again, that's clapping their lungs. Here is a pearl production. If the study, did I mention it somewhere? Yes, I did. Okay, great. Here is your mantle, and here is the nacreous layer, the most inner layer. And if there is something gets stuck right in between the mantle and the nacreous, the shell, then the animal, the mantle, has the ability to make calcium carbonate around it, and that pearl is formed, and then they expel the pearl out after a while. So this is formation of a pearl in these animals is a mechanism of defense. A sand, a parasite, a piece of a wood, whatever gets stuck there, they form the pearl. So what they do, they've studied these for centuries, and just recently they found out that if you put a mantle from another animal, it gives you the best quality pearl. Of course, farm raised. Again, the wild pearl has a better quality, it's more expensive, it's a better quality than farm raised pearl. Am I making some sense? So, but they found out if you grab this, it's a mantle, they call it nucleation. The best method is inserting mantle from another oyster right here, so it gives you epithelial cells of the mantle right here, visceral mass right there. So it gives you the best quality. So this process called nucleation. Nucleation is to put another oyster mantle in there to give you the best quality. That's what they just found out recently. I'm saying recently, I think 10, 20 years. Here is pretty much the anatomy of the animal. If you're cutting it, this is the gross anatomy on top. You have your adductor muscles, anterior adductor muscles, posterior adductor muscles. Here is the mantle. Here are 2 layers of gills, okay? And of course, food and particles are coming into the gills. Here is a sensory palp. They have a palp, they can sense the environment. Here's a foot that has not been opened yet. When you open up your shell, this is what you're supposed to see. Everything in there you see. And then when you go ahead and cut up your foot longitudinally, you grab a scalp and then you cut it this way. You cut it longitudinally. You cannot cut it any other way to see everything. When you cut it longitudinally, the foot, then these are some of the structures you should be able to see. You will see the gonads right here. All of that structure right here is gonads. But the tube inside that wiggles back and forth is the intestine. And on top, you should have stomach. Then on top right here, as I said, you have heart. Underneath, kidney. The heart has pericardium around it. So this is coelomic area. And intestine, very unusual animal, intestine passes right in the middle of the heart. Okay, so intestine of the animal goes through the heart and end up in anus. Of course, here's the gill that you saw in gross anatomy. Anything else in here, don't worry about the blood vessels, the aorta, and so on and so forth. That is pretty much what you have to know on the model right here. Okay, everything that is here except the blood vessels and everything else is, don't worry about excarnate siphon and incarnate siphon. Don't worry about that either. I don't see any that model. Here is a cross section of the foot. You do have slide of it. It says clam gill. But again, one of the slide, you have 2 sets of slide in there. I forgot to mention about radula. Make sure you look at the radula. We have 2 different types of radula in your slide collection. And the clam gill, you have one slide that just shows you the gill. The other slide, it shows you the cross section of the foot. So you see the mantle on your cross section. You see 2 layers of gills, mantle here, 2 layers of gills here, and you see the foot. In the foot, you see gonads, and of course, 2 cross section of the intestine. And on top, you will see the heart, pericardium, intestine pass through the heart, and underneath of the heart, you see the kidney. You do have that slide. This slide, I don't think is on DocuSheet. It's a new slide we got, so it's not on DocuSheet. It's in the book, yeah. All of this is in the book. Clam gill, the clam shell was used for button industry, and when I took a class in Minnesota, there were, I saw a picture, he showed up, came and the class, and he showed up some pictures on 2x2s in the class. As big as this room, there was a, how do I know it was like, there was a gentleman standing next to the pile, big pile of bees. So at the turn of the century, they took the button. I forgot to wear my clothes. Have you seen your button? Some of you, if you look at the button of your shirt, you see there is a brownish coloring into it. Have you noticed that? Some of you, I'll bring my, I do have it in your collection. I do have one of my clothing that the button has a brown line in through it. And have you ever wondered why is that? Because the old buttons or like they want to make it similar to the old days button. The old days button were not completely white. It was a little bit of brown into it because they took it from shell. So they saw that at the turn of centuries, all of the clams were going extinct because they were picking them up from Mississippi River, on the banks of Mississippi River. So they said they banned it. They said no more. Nobody can use clam for button. So that could be a lab practical exam question. I asked what is shell of clams used for, what industry they were used for, button industry. So there is the animals were going extinct. Pollution and using them for button. So they said no more, no more, and they used plastic. Thank God to plastic came. They used, here we go, they decorate you and everything else. Water current, I'm not worried about any of these guys. Sexes are separate, so on and so forth. Glucydium, we do have a slide of Glucydium added to your collection. They look like villager larvae, but look at them closely. They are different. If you look at them, they are different. The Glucydium is a new one. I don't think it's in a docu-share. The Glucydium, what happens, these larval stages attach the gills of fish and they suck blood until they are mature enough, old enough, then they detach themselves from the gills of fish and move on and become a clam. Eggs and larvae found in marsupium. Marsupium is a portion of the gill that is, these eggs and larvae can be found in, as I talked about it earlier on. Yes?
Separate sexes means dioecious?
They're dioecious. Some species are dioecious, yes. Stock reproduction. Feeding, shipworm. We do have a sample of shipworm. What happens to these guys? They attach the ships. Now ships, most of them are made up of fiberglass, but in old days, they used to attach the bottom of the ship and eat the wood. They belong to this class, Plesiopoda, and Tridio is the name of the organisms. And the common name for them is termite of the sea. Okay, so that's, here they are, look at them, this is it. They have reduced shell, and they are attached to the ship. Class of Cephalopoda, the last one. We do have squids in the lab. If you do not see it in your box, please let me know. We have mollusks, nautilus, devilfish we don't have, and cuttlefish we do not have. Those two we do not have, but the rest of them, those three we have in the lab. All marines and predators, modified foot in the head region. Archithius is the name of the giant squids. The name of the genus of the giant squid. Some do have shell, some have poison gland squids. Inside mouth they have chitinous beaks for piercing. Forms nautilus, shell is divided by septa. Do you remember? Here, I draw it. I did draw it on the board earlier this morning. These are all called septa. But the septa are connected together by, I don't know if you guys can see this, by, it's called siphuncle. I don't know if you guys can see this or not, but we do have it in the container in one of those jars. So I did draw it on the board this morning. Squids have pen. I talked about that, the internal shell. And octopus do not have shell, have 2 eyes. The eyes can form image. In squids, 2 long tentacles and 4 pairs of arms. Arms have suckers for attachment. Pen is chitinous skeletal structures which protect the cephalic ganglia of the brain. On the surface they have chromatophore. Does anyone remember what is chromatophore? Yeah, it's a color pigment that changes. The amazing thing that, again, I do not have explanation for it. I don't think they have explanation for it. How these animals recognize what is the outside environment look like. So of course they have eyes and they can see. And they can change the color to the environment they're located in. That is the amazing thing. If they could change the color only to a certain among certain type of coloration, that could be explained. But how they can recognize the outside and then change that, that's the amazing. Locomotion, it's called water from mantle cavity through the ventral siphon, lateral fins, not found in octopus, ink sac, some species, no cilia and gills, closed circulatory system, we talked about that. Reproduction sexes are separate, seminal vesicles or store spermatozoa. Hecticotylus is the name of the arm in the squids that is used for reproduction, for the male. The male transfers sperm to female by this arm. That's the name of the arm in the male that transferred female. Egg fertilized in oviduct, and malacology is the science of studying mollusks. I said that at the very beginning. Okay. We did it in a very speedy fashion.